RAFT process simulation, producing collagen-based 3D cell cultures in under 1 hour. Stage 1. Setup and incubation. The user mixes neutralized collagen with cells and pipettes the mixture into the plate well. The cell-containing collagen solution is incubated on the heater for 15 minutes at 37 degrees Celsius to form a hydrogel. The absorber is positioned and a controlled amount of liquid is removed from the hydrogel, leaving cells encapsulated in collagen and evenly distributed in the culture. Key elements of the process: absorber, plate well, cell or collagen hydrogel. Stage 2. 3D cell culture formation commences. The absorber gradually takes up liquid from the hydrogel. The process creates multiple cell cultures simultaneously, with the researcher's choice of cells with good reproducibility. Stage 3. 3D cell culture creation. As liquid is absorbed, the concentration of collagen and cells increases. A range of in vivo cell and collagen concentrations can be formed. Stage 4. 3D cell culture formation complete. The absorption is complete, resulting in an approximately 50-fold increase in concentration of cells and collagen. The resulting tissue model is strong, transparent, and approximately 100 micrometers thick. Stage 5. 3D cell culture ready to use. The used absorber is withdrawn and disposed of, leaving the cell culture in situ. The RAFT process has produced a well-defined 3D cell culture in a convenient, simple-to-use format, ready for experimentation. The user may also co-culture a different cell type on top of this tissue, or repeat the process with another layer of collagen and/or cells to form a multi-layer cell culture.